Okay. For the evolution, we need a diversity generation and then selection. And to make it better, the sexual recombination can help to make more diversity quickly. Survival of maybe fittest. It's not the strong one can survive. Survival one is just strong. You cannot define which is the strong. So, adapting to the environment and then survive, then you are entitled to be strong. Yeah, non-identical twin is similar to regular brother and sister, 50%. How many nodes between my brother and myself? Each one? Each one? How many nodes? 3천? 4천? 2 nodes. 2 nodes, then it should be 25%. Why 50%? We share the same mother and father, assuming if my father is really my father. Then it's 50%. Then that's really 2 nodes. That's a real which one? Then 25%. Queen and myself is only sharing half. Even assuming same father, same mother. Bees, the female bees has 2 ends, right? 2 sets of DNA. One set is from father, one set is from mother. Male bee is 1 end. That means one male bee can make only one kind of sperm. There's no combination. Then, if my mother, my queen, married only one male, then the sperm information is exactly the same. Half is the same. And then the other half is a combination. So 50 plus 25. Between one piece, there's a 75% I think. So that means me and my sister, work bee, is closer than me and my mother. Then, if they are in danger, who should I save? My sister. If I save 2 of my sisters, then it's 1.5 times of me. 그래서 남는 장사. If I save my mother, I should save 2 mothers. Then that's 100%. So it's a very strange or the interesting phenomena in this family. Why they are really willingly sacrifice themselves to save my sisters. That is the view of substitution. first question. Assuming that we have the same father and mother, same grandfather and grandmother. Today, yeah, it's a one case. If I can say 8 of my cousins, then it's the same, 100% of myself. Okay, there is no ACL, and T cell does not make antibody. T cell has its own receptors, and T cells make antibodies. Molecular size means molecular weight. Molecular weight of antibody is 150,000 Dalton. Quite big. Quite big. Okay, good. Many of you got the right answer. Some of you disagree. And live T cell cannot make antibody. It should be activated by antigen. Live means before it's exposed to antigen. And hypermutate it to make germline antibody into more specified. Okay, for now, since like we are at this stage, next week we will change this video. But up to today, we follow this Darwinism, Darwinian evolution. Okay, keep the book down. Okay. Okay, today we study the genetics and evolution. Genetics is a study about the genes. In which aspect? Transferring the genes to the next generation, or changing those genes. That's kind of genetics. If that generation is over and over, seems like we are not just only giving. Seems like something is changing in there. It is not exactly the offsprings are not exactly the same as our generation. It keeps changing because the environment keeps changing too. So the evolutionary law, okay, the woman will teach us about the classical evolution today. You welcome me. Okay. Then I will you are not allowed to do this. You talk, not read.
Hello everyone, I'm Dong Kim, majoring in chemistry at Postec, and today I'll talk about Lamarck's theory of use and disuse and Darwin's theory of evolution. These are contents of today's class, and the first I'll introduce the Lamarck's theory of use and disuse and Darwin's theory of evolution. And finally,
교수님, ready?
and finally, I talk about the comparison of two theories. Before talk about the theory, I'll first introduce who is Lamarck. Is in this picture, the man is Lamarck, and he's a French biologist, and also he studied medicine at a medical school, and botany at the National Botanic Garden, and then he worked a great geology in other institutions. And he also claims that theories of use and disuse in his book Animal Philosophy in 8009. Let's look closer about his theory. This is theory of use and disuse, and according to him, while living traits acquired as a result of adaptation to the environment, and we call this trait as acquired traits, and these are inherited to the next generation, and this will result in evolution. In other words, traits are used a lot, developed and inherited, and those that are not used are degenerated. For example, in the picture there are giraffe, and he want to eat the green one smaller than him. So he want to eat them, but he didn't do it firstly. But he used his neck more and more, and he will get a more longer neck, and he can finally eat the green leaf. Yeah. So in this theory, there are limits and significance. And the limits of his theory, modern genetics have shown that acquired traits are not inherited. But there are also significance, and the significance is that arguing for the first time the core concept of evolution, that life can change by the environment. And the next chapter is Darwin's theory of evolution. And in the picture, he is Darwin. Quite scary, right? And he's Charles Darwin, as we know. And he's an English biologist. And he also biologist and geologist, naturalist, and philosopher. And he also claims the theory of evolution on his book on the origin of species by means of natural selection, or the preservation of favored galaxies in the struggle for life. Quite long, right? As we know, the origin of species. He claims the theory which greatly affects humanity as well as the scientific community, and he is also a great figure who influenced most life scientists. So, according to his theory, evolution in a popular consists of three main stages. The first stage is the overproduction. A large number of offsprings that contain various mutations are produced. Each of these individuals has their own genes. And second stage is competition. They go through the struggle for existence depending on the limited resource and environment, such as food, habitat, and spouse. Right? And third stage is the natural selection. This results in a relatively increasing number of individuals with genes that express characteristics that better adapt to environment, and eventually the existing species is replaced by other species. And we call this evolution, right? And this is called natural selection. So there are also significance and limit. Significance of this theory is that it explains evolution from the perspective of species, not individuals, like Lamarck's theory. It also reveals that the driving force of evolution is not the will of individuals, but the variety of randomly occurring traits and natural selection, resulting from them. Natural selection resulting from them, the random mutation. So also evolution is a random process, so it is directionless and not a progressive process. But there are also limits. It was before DNA was discovered at the time. So therefore it was not possible to explain the cause of mutation and the genetic principle. So finally, I'll talk about the comparison of two theories. There are two main differences between the two theories. First of all, in the case of Lamarck, they think that changes that occur after birth are inherited, but in the Darwin's theory of evolution, I think that traits that were possessed at birth are inherited as they are. In addition, in the case of Lamarck's theory, it is thought that evolution occurs by changing and inherited traits according to the will of each individual. But in Darwin's theory of evolution, it is thought that evolution occurs due to random mutation in species and natural selection. And there are also some experiments that prove the Lamarck's theory are failed. And let me explain about the experiment. So in the experiment, scientists cut the mice's tail. Scientists repeated that cutting the tail of mice and mate each other to give birth and make other offsprings of short-tailed mice. So they repeat again, and finally there are offsprings which has short-tailed mice parents. But finally, scientists measuring the length of mice's tail, but there are no changes of the length. So it proved that the Lamarck's theory is not correct. And there are also, after Watson and Crick and Rosalind Franklin proved the structure of DNA structure, the modern biologists revealed that there are DNA and there can be mutated by other things. So it also proved that Darwin is wrong and Lamarck's right. So my conclusion is that Darwin is correct. So thank you for listening, and is there any question?
Is there any question?
According to the Darwin theory that
Don't read. Watch him and talk.
according to Darwin's theory that it is explained that the fittest one is the strongest one, the most highest percentage of which can survive. But some animals like peacocks or budgerigars, they have very fancy or very beautiful feathers, and having these feathers is very big prices because many predators can attack them because they can identify, they can see them because very they have very colorful hair, colorful feathers. So can you explain how these phenotypes can be survived even though they can be easily attacked by predators?
Oh, that's a good question, and I think that although the people can be more dangerous if they have more fancy feathers, but I think they can also have good, the more good, the better chance to mate other spouse, right? And I think in the experiment of nature, they can survive since can more they can have more chance to mate other people. So I think it has more profit to have feather.
Okay, other question? Okay, thank you, Don. 인터뷰 친구는 아니지? 오케이, 잘했어. 오케이. So based on this, now the reason why the Darwinism is correct, we thought, or at least in my generation, up to high school, in biology class, we learned Lamarck is wrong. How about your case? In high school, did you learn Lamarck is right and Darwin is wrong? Maybe it's kind of a changing situation. Now the key part is the gene is stored in DNA. That's kind of observation we found. Last century we found. Still we believe that genetic information should be stored in DNA. And whether this DNA can be transferred to the next generation, that's the key. Even though you cut the tail, that information is not stored in DNA. And that information cannot be transferred to the egg cell or sperm cell. That's why Darwin is correct. It's the molecular level of explanation. Okay? DNA information is a digital information, right? Not binary, quaternary information. 3-bit make one amino acid, 3-bit computer. The quaternary computer system, it's a digital information. If you don't change that code, that information, the tail cut information cannot be stored there. That's the implication why Darwin is correct. Molecular level, Lamarck is not correct. Now what we hear, the picture in the Facebook layer, if they train this hand so much, then eventually they will be like this, right? Not like me, it's getting longer. Whether this character can be transferred to the offsprings or not. Maybe not. Okay, we will see. Okay, so far we believe that kind of trait cannot be used, but according to the Young search, root word is used whether it can affect the next spring or not. Okay. So in the evolution, at least we know that our next generations are not exactly the same to us. Similar, looks like us, but not exactly. And direction of evolution, still the one kept saying that the strong ones survived. There is nothing defined as a strong. The direction is just a survival one. Is the fittest, and that is sometimes we call they are strong, but there is no definite definition of a strong to survive. Okay, sometimes the too strong, too brave die first, right? Then it cannot be strong one in terms of evolution. And peacock case, they have a very funny, very big, the beautiful the tail, and that is seems like it's demonstrated that I'm strong enough. Even though I use my money, my energy for this, still I can survive better. If that is true, then he has a better chance to mate and make more offsprings, more babies. Then, in terms of evolution, this kind of survivor or winner. Okay, that does not mean that this feather is really make the peacock really physically strong. Maybe not. Okay. So now the evidence of evolution, whether that's really happening or not. If you are the Christian, maybe, this is creationism should be the truth, maybe. But in real world, we see that evolution happening all the time. So this is a post taken 5 years ago, just before the corona situation. And we live like 30 years is one generation, usually we say. This is 30 years ago. So I'm one of them here. So that's 30 years ago. 파치라픈, 이탁식. And 30 years ago, now that they look like a different shape, their head is becoming bigger and bigger. And another 30 years ago, even bigger. So these are the Koreans. Every 30 years, they keep changing. The shape and also the face, they maybe similar, looks like us, but they are quite different. Over just 100 years of time, we will keep changing, following the environmental changes. Okay, evolution is happening. That's what we observe every day in the laboratory. Okay, so for the evolution, one component for evolution is we need to make diverse offsprings. To make that, all the information are stored in DNA, digital information. Only way is we change G into A or T or the other unit. Then one way is the mutations, change one by one. Maybe that speed may not be very fast. If it's a DNA, even more stable than RNA. RNA is quite unstable. That means RNA has a better chance to change it. That's why coronavirus is an RNA virus. Their genetic information is relatively unstable. That's why they can change quicker than DNA-based virus. They are unstable, but at the same time, they can make more variations much quicker than DNA-based virus. For long term, which is more the profitable, it's not clear. Okay, if they are isolated, then with the RNA virus maybe die out quicker than DNA-based the animal life. But fast mutation is the one way. The other way, if you just make exactly the same couple of us, mutation is one way, and increase those mutations. What is the experimental or the laboratory technique to increase mutations? If you are a biologist or a geneticist, you wanted to increase, you don't want to wait 100 the generations of a fruit fly. I wanted to increase higher chance of the mutation. What do you do? Again? UV or X-ray, very strong energy. Light is very condensed, high energy. The wavelength is shorter, the energy is higher. Okay? X-ray or UV you radiate, then they can attack directly on DNA molecule, break it. Then during those repair, sometimes that signal is changing. That is artificial mutations. That's one way. But if you do too much, then too much mutations, no, I cannot survive. Or the sperm cell or egg cell cannot survive. So you need to have some range. Okay? Now in nature, they developed new strategies, so-called sex. In our case, we have two sexes, male and female. Some lives, they have multiple sexes. Anyway, using our information, now we change them. Before we count the advantage of this, let's think about this. I was the planning to give you this the last lecture, but we didn't have a chance. Computer system. Information, how much information we can store? If it's a 2-bit computer, means 2 units generate 1 word. Then how many information can you store into it? How many different kinds of information can you put there? 00, 01, 10, 11, 4. If it's a 4-bit computer, 16. If it's an 8-bit computer, 2, 5, 6. Okay, now we are getting some general rule. If it's an n-bit, then 2 to the n, right? So this bit information can be stored in there. Right? So we have the DNA case, it's a quaternary, and then the unit then 4 to the n case. That's the information we can store. So now, if it's a 2 to the 10, how many? 102424, that's approximately 1,000. Okay, if this is a very big number, then if you encounter 2 to the 10, okay, that's about 1,000. 1,000 is a 10 to the 3 kilo. Okay? 10 to the 20s? I already told you. It's a million. Okay, it's about million. So this is a unit we studied in Western culture. 3 unit: kilo, million, billion, trillion, kind of things. Okay, that kind of to simplify. Okay, now let's try to think. I showed you this in the video clip. In our genome, we don't give whole chromosome in 1 unit. If there is only 1 unit, then we can make only 2 kinds of sperm cells, right? If it's every chromosome is only in 1 unit, then we have 2 copies. Okay, 1 copy here and 1 copy here. We can make only 2 sperm cells or 2 egg cells, right? But we divided those information into 20, 30 different units. Now, when we make sperm cell or egg cell, the combination is randomly choose 1 of each. Then, how many different kinds of sperm cell or egg cell can make? It's a binary, 2 to the 23. How many is that? It's just a practice. 2 to the 20 is a million, and 2 to the 3 is 8 million. Okay. Approximately, we can make 8 million different kinds of sperm cell and egg cells. That's by simple combinations, we can generate another values. All of a sudden, when we need to make exactly the same copy, just a point mutation we should try, that's not so efficient. Now if we have this number of divided information, we shuffle them, okay, and we can generate 8 million different kinds of options. Now, this question, if you are not identical the twin, okay? So my brother and myself, or my other sibling, my sister and myself, and what is the probability of we get by chance 100% identical information from my mother and father? Then first information is, then okay, this is good. Now if the question does not include all the information, then you can define it. Okay, if I'm a woman, okay, then I don't make sperm cell, I make egg cell. Kind of the defining you can do, and then move on. So we just know those numbers. 2 to the 23 is the combination number, right? If it's combined, sperm and egg combined together, then it should be multiplied, and then it gives like this number. Okay, now what's the number of this? 2 to the 46. Second. This was 8 million, right? 8 million by 8 million. That's this, right? What's the number? Correct? This one? Right? Then? 64 trillion. Okay? So, usually if you encounter this kind of a big number, we use log value or exponential value to make it easy to move that number. But now here, if you see here, it's important to convert this quickly to this kind of common the daily lives of the number system. So this is mega, giga, tera, 64 tera combinations. Then probability is 1 right over here. This is probability. By that chance, I can have exactly the same information like identical twin with my siblings. Okay? That means the combination, one couple of male and female of a human can generate this many different babies. That's why having almost the same babies is quite difficult. Using that possibility, now by developing this sex system means recombination system. It's more difficult rather than just I make my own copy. I need to find my mate, my partner. And sometimes it's quite difficult to find the partner. Still, overcoming that hurdles, still it can generate a lot of the advantage in terms of diversity. So for making healthy surviving chance for evolution, diversity is very important. Because once the environment changes, homogeneous monoclonal society is very easy to go to the everything dies. Okay, another question. My uncle and me shares 1%. My uncle and me, uncle is a samson, right? 3 nodes. So that's 1 8. 1 node is a half. It becomes quarter like this, just because we have same mother and same father. That saves the double of those possibilities, so that's a 1/4. Okay, not only those, the combination of different chromosomes, even chromosome itself can change some part of it. Now, originally this is the father side, this is the mother side, let's say. When they make its the father side sperm, it should be 1 unit and the other unit. But sometimes when they meet, both of them are father side. First of all, they make same copy, and then they exchange some part of the gene, even chromosome to chromosome. Then the diversity becomes even higher. Not only this originally we counted this as 1 unit, but now it can generate 1.5, 1.7 kind of units. More diversity can be generated by this kind of recombination. In this case, we call homologous recombination. Utilizing this homology, and then some part of that can be changed. Okay? That can be used for genetic engineering later too, to replace some part of information with different genes. Okay, antibody and immune system. Antibody can be generated by B cells, and then when B cells mature and then meet the antigen, it generates antibodies. Usually millions of antibodies can be generated to make those the generally build antibody. This kind of a Y shape of antibody structure, altogether 150 kilodalton. And if you look at that, the protein is immunoglobulin, and total size is about 150. Now if you look at carefully, it has a 1 red unit, 1 blue unit, 1 yellow, 1 green unit. Those are separate proteins, and they are assembled together. So that means approximately this one and this one is the same size. Now if you look at here, this is heavy chain, long one. This is light chain, shorter one. Then altogether this is 150. One heavy chain, altogether 150. One heavy chain, about 50. Okay. One light chain, about half of it. It's easy to memorize. But the total number you need to know. So using this, now this is a constant unit. They don't change. And upward here, they have antigen binding fragment, this part. So antigen binding, this is a heavy chain, this is a light chain. Antigen binding site is here. Okay. Now, FC, what's the size? Constant solution. Constant or the crystallizable, they say, but it doesn't matter. And it's the same structure here. It's kind of just a holding part. What's the size of a constant solution? 50. About 50, okay. and FAB about 50. Now, the binding site here, this site is called as a paratope. All other regions are the same, only this part is different. That differences come from heavy chain and light chain together and make this kind of pocket. That's a paratope. In contrast, in antigen side, so this is antibody. I'm recognizing genome. Okay. Which part I recognizing him? That depends on the situations. If genome is injected into my body, I make antibody. Only one antibody? No, I make maybe thousands or millions of antibody against the genome. That means it can bind to different sites of the genome. Okay, then the binding site on genome. This part on genome, that is called as epitope. Even though genome is a big antigen, genome is a big protein, but the binding site is not so big. It's only 5 to 6 amino acid size of a small region. Okay? That is so-called part of the antigen, which is recognized by antibody. That is epitope. The partner site on antibody is called the paratope. Okay, paratope is here. Epitope is a partner of paratope. Okay, paratope binds to antibody. Now, based on this, so far we learned classical definition of the genes. One gene make one protein, and we use mRNA as intermediate information. Then we said the antibody can be generated many, maybe millions of antibody. Then how many genes do we need? In genome sequence, the result, how many genes do we have in our body? 25,000 is kind of maximum number we have. Antibody number, millions. Then where do we store that information on DNA? We already studied in the video clip. We don't make them all, we just make the constant part only 1 unit. The variable part, paratope side, we save those informations as a fragmented information. Among them, we take each of them one by one out of here, and then it can make this kind of a combination. That combination can make 8,000 from heavy chain, and in this case, 200 from light chain, and they meet together, and then eventually they make this paratope. It can generate 1.6 million. Huge. How many genes we kept on the gene? Could you count quickly? Now, rather than multiply, we just add up those. Okay, then if you add up, here 84, here 45, all together, we just use 130 genes stored in DNA. That means in the beginning, it has all the combinations. When they develop into the mature form, they select only one copy each, and then make one clone each. Okay. Utilizing 130 gene by using this combination power. This combination comparing to the sperm and egg cell combination, much higher efficiency. In egg cell and sperm cell, we took out of 2, we choose 1. In this case, out of 50, we choose 1. Out of 27, we choose 1. That's why we can generate a lot of combinations. That's how we can develop huge numbers of antibodies. They are not perfect, but this is a starting point. Germline is the cells which make a sperm cell or egg cells. That means this information is we got from father and mother. Means this is one cell. And another cell, if this is the father side, then we have another cell from mother. Then we have 129, and the other one is maybe another 130, kind of a gene cell. And altogether, originally this could make 1.6 million. Then the other cell also can make about similar number. Altogether, we have 3 million. Interestingly, my germline antibody and your germline antibody are all different. And the starting point is different. That's why sometimes some of you you can survive better against the COVID. I have more suffering because I don't have fitting very well to the virus. Okay, germline is starting point is different. And in the beginning, the germline antibodies, we don't know what kind of enemy we will see. Okay, what kind of the virus, what kind of antigen we will encounter. That's why in the beginning, those germlines are not very specified. And that's the term of Kian Lock, and this is the date model. And many of you got the right idea, but still some of you has a little mixed idea. So I want to point out. In the beginning, the germline antibodies are flexible. That's why it can bind to this shape of antigen or different shape of antigen like this, because I can change my structure. And even this kind of structure I can bind. Once it binds, this shape and then this shape is different, and then I keep changing its positions, and then end result is hypermutated antibodies like this. Doesn't move much. Okay, this is key and lock. First one is induced fit. In the beginning, because the binding affinity is a little lower, they can bind many different things to increase the chances. Later, if it's hypermutated, then this one can bind to this one very strongly, but now it cannot bind to this anymore. Okay. This is the process of with the development antibodies in our body. That takes how long? 1 2 weeks or 3 weeks. Okay. So when we get those antigen first, we don't develop antibody right away. We should survive for 2 weeks until our body generates antibody. Okay. After that, next time if we get the COVID, we already have that antibody. Then we can boost it right away in 1 day. We can generate antibody and antibody. Okay. I'm not sure whether I told you this or not. Long time ago, if we are bite by the snake, it's the poison. It's a very urgent situation. We want to neutralize it, but it's difficult to get the antibody for it. We need 2 weeks, but it can kill me in 3 minutes or 1 hour. In that case, the people have used the snake poison, snake venom, injected into horse, and then collect horse serum that contains the horse antibodies. It can neutralize the venom. So if you are in emergency cases, you get that shot. And that's polyclonal antibody. So it's not selected, it's a mixture of antibody, not monoclonal. But it can neutralize that venom. Then you can save your life. You cannot use that antibody again. Why? So next year, I got bitten by the snake again. If I injected that same the horse, the serum, what happens? My body already developed the antibody against the horse antibody. Right? First time it was okay, because it was too early, and then we are ready for next time. But the next time, if you get the shot, then now you will die because of the antibody response, so-called cytokine storm or over the immune system. In that case, what's your choice? Okay, then evolutionary he lost. Who is the evolutionary winner to survive? What is your choice? Now you take cow serum. The next goat serum. Because each the animal's antibody is different. So horse serum against horse serum, I developed antibody, but still I'm okay for different immune system. Okay? So that is the antibody system. It takes time to respond to antigens. Okay, molecular evolution. Evolution case, the unit of evolution is individual, like us. Okay? If you cannot survive, then we don't have a chance to transfer our genetic information to other species. If we mimic it, only for the gene part, we said In evolution, we have mutation and selection. Mutation is the diversity generation, and selection is the selecting the winners. And as another strategy of mutation, maybe recombination can use too. In this case, those information should be stored in DNA. Then it can be transferred. And classical size of evolution, individual like us, are the unit. But it can be cell level too. We just have seen those B cells. B cells are selecting to the survivors. That's kind of a cellular level of evolution. Out of 1.6 million combinations, only winners are responding best to the antigens. Then they can be survived. They are kind of a winner. It can go down. Maybe if you think about only that antibody making gene, then we don't need a whole cell. Just we need antibody gene or protein. That is the unit of surviving, right? Similar to the surface gene case. Maybe we don't need the whole body. We just need only surviving gene and then combine. Then we can make up kind of a survival machine like us. Now mimicking this, then forget about the individual or forget about the cell. We just count protein and then its information, DNA. If you combine them, then maybe this evolution we can mimic in the laboratory. That's what we call molecular evolution. We take DNA and let it make a protein, and then follow this process of selection. One example is not fully molecular, but similar to molecular, is using virus. Virus's role is just surviving and then transfer those DNA multiplying. But here, it will insert some specific gene and it will make a protein. That protein can be expressed either on this surface, P8 protein, or P3 protein on this surface. Here, it can be small protein. Here, it can be much bigger protein. We can incorporate. And then, by selection, so if we make protein here, and then utilizing any binder, and we can select the best binding protein from virus mixtures. Virus is very small. What is the size of the virus? Chicken egg is like 10 centimeter. Human egg is much bigger than regular cells. Regular cell is 10 micrometer. Human egg cells 150 micrometer. If you have good eyes, then you can see with the naked eyes. That's it. And virus size? Human cell 10 micrometer. Bacteria? 1 micrometer. Virus? 0.1 micrometer. 100 nanometer. That means 1 micrometer versus 100 nanometer. Size is 10%. Volume-wise, 1,000 times smaller. Right? It's a three-dimensional. That means in 1 liter, if you have a virus, bacteria 1 million, then same volume you can have 1 billion virus in there. Okay? Then this is quite small machine which make protein like this. And let's say you have some target. Okay, this is virus protein, coronavirus protein. So you put this and then put into the mixture which bacteria, which virus binds to this. Then this peptide can be good candidate which can bind to coronavirus protein. Right? You generate billions of it and then you select one best one. Quite attractive, isn't it? In principle, compared to antibody is million we are playing with. And hypermutation. With this phase display, very small, much smaller than B cells, 100 times smaller. B cell is 10 micrometer, virus is 0.1 micrometer. 100 times smaller means million times by volume, right? One B cell can do, then million virus can do. Much higher diversity you can generate. Okay, so this is the power of phage display. I asked you some questions. What's the advantage and disadvantage of this phage display? Phage is one kind of virus which can but in fact bacteria. Advantage is it can make very rapidly generate huge diversity. Easily it can reach billion. So diversity wise it's much stronger. Problem is it can only interact, it can be selected by binding acid. Virus particle is generated, you add the bait and then check which one is binding to. That assay system is limited only to the binding event. That is kind of the limitations. Sometimes we want a little more diverse effect. By adding this and whether this virus can kill the cell or not. Kind of a functional assay is what it is. But in this case, only binding event you can measure. That's kind of the limitation. Also, I asked whether is there any better, more advanced one. Well, maybe some of you will catch it. Ribosomal display. Virus is 100 nanometer. It's quite small, but if you can isolate out that DNA, the one DNA, and this is your protein. If this can make protein right away. Now this is one molecule. Size of one molecule, it can be maybe 1 nanometer. Now, 100 nanometer of virus, phage display also quite high diversity. If you can reduce it down to 1% the size, then diversity becomes 100 million times, right? So this is ribosomal display is the one example like that. How do we? mRNA can make protein, so you make mRNA library. Mix that together with the ribosome. Ribosome is a machinery which translates it into protein. And ribosome read this information, make protein here, and don't let them leave. You can modify, you can engineer, and then this information carries this protein together. Information and machinery connected together, and number-wise million times higher. Now trillion. In one small reaction vessel, you have trillions of different proteins, and you do selection. Now you need to amplify this mRNA. What is the technique to amplify the gene? PCR. The problem is the PCR is template is DNA, product is DNA. Now your information is RNA. You need something more. Reverse transcription. RNA viruses, they developed some other non-common enzymes. Usually our genetic system, DNA to DNA, DNA templated to DNA synthetase, right? DNA templated DNA polymerase. To make RNA, DNA templated RNA polymerase. The next step is ribosome. Now, RNA viruses, sometimes they need to make a DNA from their genome to insert that information into the host genome. They developed something funny, RNA templated DNA polymerase. That kind of enzyme we call reverse transcriptase. Transcriptase is a DNA to RNA. This is reverse way, this is a different enzyme. So if you use that enzyme, then you can change RNA into DNA, and then you can use PCR. Then you can amplify this. This is mimicking the life process. So this is the ultimate level of molecular evolution. Now this kind of system is now not only in the biological system, now they use this for chemical synthesis too. You add chemicals here and then encode that information on DNA, and then drug candidate, you make millions of them or billions of them and select the best drug compounds. That's also over. Okay, so we are here right now. We are 2 weeks away from midterm exam. Already, right? Yeah, too fast. So today we started about evolution, and Darwin was correct. Next week we are going to talk different story. Darwin is not always wrong, always correct. There's another mechanism. I picture my some can be good picture too, kind of a story, in terms of epigenetics. Okay? 동원이가 짜진 않았는데 동원이가 딱 볼스 스탑 해줘서 좋았어. 꼬리 자른 얘기가 나왔을 때 혹시 에피제네틱스 욕을 하면 안 되는데 잘 스탑했고. Next week we are moving to different story. Now, so I'm inviting the presenters. So far we have antibiotics, vitamin, chemotherapy, 대응이, still open slots here. You may choose your preferable the topic. First come, first serve. 선착순. So next week is still open. You can send me 카톡 or email, and still it's open next week. Epigenetics is a very charming topic. The book, first book, 유전자는 네가 뭔 짓을 했는지 알고 있다? Is the epigenetics story. That's one of those the cutting edge, new the idea and the new the science. It's worth trying to spend some time and read some of the materials and include them. Okay? Open. You can volunteer. First come, first serve. Any questions? Okay.
With the evolution, you were saying that it's still happening now, but what's the selection pressure now? Because like when you go to like if you're like allergic to peanuts, for example, there's medicine that will keep you alive when as before you would die and then not pass on the allergy, but now you can pass that on to your children, which like shouldn't necessarily.
Right. If you think ourselves as the subject of a study, it's like 80 years, maybe 100 years of lifetime, and die or not, then maybe live too long. Still, we can think hypothetically. Let's say if somebody has the allergy to the peanut, they die out, then maybe other populations they are more resistant to the allergy to the peanut. Maybe that's more advantageous. Maybe our main food is only the peanut is left over. Without it, we cannot survive. Then those populations are not eating very well. So that's kind of an example of the evolution still ongoing. Now, if we can reduce that into the laboratory size, then we can make those bacteria or virus. It can be done within 1 week or 1 day. Then we can see whether how the evolution is moving on. That's kind of the laboratory size, but in real life, we always see antibiotic resistance. That's typical example of evolution here. Then on this week, we are going to think a little more details of how they change and adapt into the more better fitting the cell. Okay, other question? 한소리가 질문을 했는데 시간이 되지. Okay. I said the three scenarios of protein synthesis. Maybe in the video or on the class, I said a little confusing way. Let's re-summarize. So 3 cases of location of protein synthesis. One scenario is ribosome is in cytosol. Protein is synthesized, and the product is in inner world or outer space. Say again, ribosome is in cytosol, inner space, and then product was generated in the cytosol, and the product is the inner space or outer space. That's inner space. If the product is the ribosome is attached to the ER circuits, product is inserted into lumen of ER and is secreted into the lumen of ER. Then in scenario 2, what is the final location of this product? That's totally outside of the cell, right? It's moving to the outside of the cell. Through exocytosis, product is secreted to the outer space. So the case, this is synthesized into the lumen, but at the end, it is not secreted, it is anchored on membrane, the ER membrane. Then, majority of the part is in outer space, right? Then, what can be its end location? If we go to the plasma membrane, facing inward or outward? It's outward, right? Now, this is the basic 3 scenarios, but there may be more variations definitely. So, first scenario, if it is synthesized, and then majority part of them is in cytosol, and end part, if it has some binding motif or signaling on the peptide, if it binds to the ER surface, let's assume. Then, if this moves to the plasma membrane, then this is a plasma membrane protein facing outward or inward. That should be inward because majority part was in cytosol, right? It's moving outside and then cut there. Or this final sequence has different destination, like mitochondria or lysosome or different motif it has, and another shuttle will come and bring it to those membranes. Then mitochondrial protein or lysosomal protein facing inward or outward, facing into cytosol or outer space. Lysosome synthesis mainly done into cytosol. Only last part is a signaling peptide. Okay, then it can be carried to lysosome or mitochondria, and it's binding to here. Then main part of the protein will see cytosol side or inside that organism. It's a cytosol. Right? So that's kind of the general pictures. Of course, there are exceptions and different cases. That's a simplified version of the thing, how they move to different regions. Okay, any other question? Okay, if not, next week we are moving to another very interesting topic. Topic is quite exotic. That's why I didn't put very long video clips. But the content is used in 20% of the time. 숙제도 안 줄 테니까 다음 주에는 여러 가지 방법으로 the following your own way try to study about the epigenetic. That's quite easy. Okay.